Hello, my name is Amias. I will be teaching infectious diseases today. Um, okay, so lesson outline for today. So we'll be covering content, how to memorize effectively, I guess, the big chapter of infectious diseases. So when we talk about effective memorizing, we want to talk about looking at all the small little pictures, so each subtopic within the infectious disease topic, and then making it into one big picture so that when you think of infectious diseases, you have a idea of how each individual component links to the other. Okay, so the small versus big picture, we want to see how everything adds up. Okay, and then we're going to cover a typical infectious diseases question, and we're going to talk about inter and intra topic links. So we're going to be talking about how um, like a specific part of infectious diseases can be linked to proteins or other chapters that we have. And then if you have any questions, you can just type them in the chat. And a preface for the whole lesson today is that different schools have different keywords and different terminologies that they use. So if anything here is foreign to you, you can stick to how your school teaches you. And yeah, and if you have any questions as to what certain terms mean, you can just type them in the chat. Okay. And today we'll be following RI syllabus because that was my syllabus. Okay. So we're gonna talk about the learning outcomes of infectious diseases, and this is how you should study bio. So you look at your content, go back and refer to your LOs, and then see if what you have studied already, and then see if you can answer the questions with what you have studied already. So we're going to be talking about the specific and non-specific immune system. We're going to be talking about active, passive, and naturally acquired as well as artificially acquired immunity. And we're going to talk about B cells, T cells, APCs, antigen presenting cells, memory cells, how they function in your primary and secondary immune responses. Then we're going to talk about how the structure of antibodies relate to their functions, and we'll use IgG, immunoglobulin G, as an example, because that's the whole focus of the syllabus. We're going to talk about generation of antibody diversity, so that's somatic recombination, somatic hypermutation, class switching, and then, but today we're not going to be talking about vaccination because it's very, very self-explanatory. So vaccination, the use of vaccination, the benefits and risks, we're not going to be talking about. And we're also not going to be talking about influenza and HIV because your school should have covered that under viruses. And we're going to talk about the TB, Mycobacterium tuberculosis, how it is transmitted and how it infects your body. We're going to also talk about antibiotics, and we're going to focus on penicillin because that's the focus of your syllabus. Okay, so we'll start with these things called antigens and epitopes. So the difference, some schools use the terms interchangeably, and you can understand why, I guess, now. So antigens are foreign substances that induce an immune response, but epitopes are parts of an antigen. So they're not the entire antigen, they're part of the antigen that binds to the ABS, the antigen binding site of your antibodies, as well as your T cell receptors. So this is a simplified picture of how your antigen and epitopes look like. But then this is not reality because in reality, it looks like a 3D polypeptide chain, right? So this links back to your chapter on proteins. So you visualize and you have to understand that antigens are not blocks, but they are actually polypeptide chains, right? So you can see that this part here, this part is the epitope, but the entire portion here is the antigen. So, and then they bind to the T cell receptors of your antibodies, like the one in the picture, as well as your T cell receptors. Okay, and then, so, okay, and one antigen can have more than one epitope. Again, if you look at this two pictures, you can see how one antigen has more than one epitope. So next, talk about the immune system. So we have the innate and adaptive immune system. So the innate immune system is the non-specific immune system. And it's non-specific, you can think about it this way, because you have just encountered a new pathogen that you do not know, that you have never encountered before. So then you just want to defeat that pathogen, you want to get rid of the pathogen. So you have to use a general mechanism to get rid of it. So it's non-specific. It's your first line of defense. There are two portions, the anatomical barriers and the cellular components. So the anatomical barriers, we have physical and chemical barriers. And then an example of physical barrier will be your skin. Chemical barrier will be your mucus secretions or your antimicrobial substances in your saliva, something like that. Okay, so you use the examples that are given in your lecture notes if they ask. Then we have the cellular component. We have your macrophages, dendritic cells, and neutrophils. So we usually only talk about macrophage and dendritic cells because they are your antigen-presenting cells, and that is a big process in infectious diseases, and that's why it's the focus. So for your adaptive immune system, also known as your specific immune system, you have your cellular components, which include your lymphocytes, and you have your humoral components, which is basically your antibodies. So for lymphocytes, we have 3 or 2 different types of lymphocytes, and they are your naive, your effector, and memory cells. Why I say 2 or 3? Some schools classify memory cells under effector cells. Okay, and that's why some people think of it as 2 instead of 3. Okay, so your naive cells have never encountered your specific antigen before, and your effector cells are activated by your APCs. So your naive cells, after they've been activated, become your effector cells, and your memory cells just help you mediate your secondary response to the subsequent encounter with the same antigen. And you have to use the word same when you're talking about secondary response, because if you don't use the word same, you won't get the mark, and then you won't score. So this is also something you have to know: the derivation of your B and T cells. So they develop from your hematopoietic stem cells, and then they differentiate into your B cells and your T cells, and B for bone marrow, T for thymus. So your hematopoietic stem cells, if they differentiate in your bone marrow, they become your B cells. If they differentiate in your thymus, they become your T cells. Okay, so we're not going to talk much about your innate immune system because that is very self-explanatory, and we'll talk about antigen presentation. Okay, and so antigen presentation just refers to the display of peptides of an antigen bound to your MHC proteins. MHC stands for major histocompatibility complex. So these are on the and they will be displayed on the surfaces of your APCs. So your step 1 will be your formation of pseudopodia, and some schools do not include this. But so pseudopodia is just these finger-like projections you see here. So that's your pseudopodia. So the formation of your pseudopodia, so your phagocyte will engulf your antigen and then form a phagosome. If you look at the picture, there's the number 2 here. So the phagosome is what encapsulates the antigen. Then after the phagosome fuses with your lysosome, it forms a phagolysosome. Here, step 3. And then inside your phagolysosome is where your antigen gets digested into your small peptides. Okay, so this is the digestion part. And then the step 5 is where your peptides bind to your MHC proteins and form your peptide MHC complex. Then they're transported via vesicles to the surface of your APCs, and then that's how your peptide MHC complex is shown and presented on the surface membrane of your APCs. And then lastly, you have your binding of your peptide MHC complex to the specific T cell receptor on your naive T cell. And this is naive T cell. You have to use the word naive to show that it has not encountered the antigen before, so that you can differentiate between your naive T cells and later on your effector T cells. So here we have the whole adaptive immunity. So after your APCs have presented their antigens, your naive T cells get activated. Okay, then they secrete cytokines, or some lecture notes refer to it as chemical signals. So cytokines, and then this will trigger clonal expansion and differentiation. Some of you may not use clonal expansion, so they use mitosis as well. Clonal expansion is just mitosis. Okay, and then they differentiate into your effector cells and your memory T cells. Okay, so this is how it looks. So you have your epitope that's recognized by your APC. APC is your macrophage and dendritic cell. Then you form your peptide MHC complex here. You present it to your naive T cell here. and then you activate it and you give your effector cells and your memory cells. Then we'll talk about the helper T cells. We'll talk about the cytotoxic T cells, sorry. So the role of your cytotoxic T cells is they secrete two things. They secrete your perforins and they secrete your granzymes. So perforins are just molecules which help to make pores in the membrane to create tiny, tiny holes in your cell membrane for your granzymes to come in. So your granzymes come in later and they diffuse via these pores and they activate enzymes within the cell to trigger apoptosis. So apoptosis is cell death. Okay, and then we have helper T cells. So the only role of your helper T cells is to activate your naive B cells. Okay, so we have this, go back here. Okay. Okay, so this is so the process of the activation of your B cells, your naive B cells, occurs in two steps. So the first step is where you have your epitope, again, the epitope of your antigen, recognized by your B cell receptor, and it's the specific B cell receptor. So it's recognized here. And then after that, it goes through a process similar to antigen presentation and in your antigen presenting cells. So your antigen gets taken up, gets digested, combines with a MHC complex, and is displayed as a peptide MHC complex on the surface of your naive B cell. Okay, so that's the first step. Your second step is where this exact same peptide MHC complex gets recognized by your helper T cells. So if you think about it, the peptide MHC complex on your naive B cell after the first stage of activation, so this one here is the exact same peptide MHC complex as this one here. It's the exact same peptide MHC complex. And because it's the exact same peptide MHC complex, the T cell receptor, the specific T cell receptor of this naive T cell is complementary to both peptide MHC complexes, the one on the APC and the one on the naive B cell. So that's how your second step of activation occurs. That's how the helper T cell recognizes and binds to the complementary peptide MHC complex on your naive B cell to activate it. So I repeat, it is naive B cells are activated in two steps. The first step is when the epitope gets recognized by the specific B cell receptor on your naive B cell. And then, okay, let me just show you this part, then it'll be easier. Okay, so the first step is where your antigen binds to the specific B cell receptor. And then the second step is where your helper T cell binds the peptide MHC complex on the cell surface membrane of your naive B cells. Okay, then again, your helper T cells secrete cytokines, which then stimulate your clonal expansion and differentiation again. Okay, and then after differentiation, your naive B cells become two things: your plasma cells and your memory B cells. So your plasma cells are the ones that secrete your antibodies, and for the rest of the lesson today, we'll be referring to both of them as effector B cells. Okay, so if you want to take a screenshot of this, you all can take a screenshot of this. Okay, so then we want to talk about the characteristics of adaptive immunity, and we want to contrast that against our characteristics of your innate immunity. So you talk about adaptive immunity, the first characteristic that you have is your high level of specificity. So if you want to give an example of your high level of specificity, you can talk about how B cells, your naive B cells, are activated in two steps. So both, if the first checkpoint is not met, it will not even go to the second checkpoint. And both checkpoints have to be passed before your antibodies are secreted, because you don't want to waste resources producing antibodies that are not complementary in shape to your epitopes on the antigens on the pathogens that are in your body. I repeat, because you don't want to waste resources creating antibodies that are not complementary to the epitopes on the antigens on the pathogens. Okay, so that is the first characteristic of adaptive immunity. It is very specific. and then we compare that to our innate immunity, our innate immunity is not specific at all because we are just whacking, okay? Because we don't know which pathogen it is. We just know it's a foreign particle and we just want to get rid of it ASAP. So there's no specificity or very low specificity, okay? And then the adaptive immunity, we have adaptive immunity shows memory because memory B and memory T cells are created, they're formed, unlike in your innate immunity where it is not formed. So there's no memory in innate immunity, there is memory in adaptive immunity. So there were some questions that came in on how to better understand the concepts of infectious diseases. So it is a really, really big topic and it's really daunting to start, I get. Okay, but then you have to look at the different links between your topic within your topic so that when you answer the questions, when the questions come and then you look at the question, you immediately can tell, okay, the characteristics of adaptive immunity and innate immunity are these, and these are the differences. If they ask you for differences between your adaptive immunity and innate immunity, then the next part of the question, because they're always linked, the next part might ask you to describe the adaptive immunity. So when you study this topic, do not study it in more than one day. Try to take it as one day, and then you see the links between your innate immunity, your adaptive immunity, and how everything comes together, and then you can better understand the topic. That's my advice. Okay, and then just try to make these links by yourself. Okay, so next we're going to contrast the primary and secondary response. So we're going to talk about the characteristics of your primary response. So in case you are a bit lost, your primary response will include your innate and adaptive immunity, and your secondary response will also include your adaptive immunity. Not so much of your innate immunity, more of your adaptive immunity. Okay, so the characteristics of your primary response is that there is a lag period because you require the clonal expansion and differentiation of your naive cells. And then you have to differentiate them into infected cells, and that all takes time because you need time to recognize that there is a foreign pathogen. And then after you recognize, you have to go through the entire antigen presentation part, and then go through clonal expansion and differentiation of your naive T and your naive B cells. Okay. The second characteristic is that there is a gradual increase of your antibody concentration, a gradual increase. And then the next one is the lower peak concentration of antibodies. So the peak of your antibody concentration will be lower in your primary response compared to your secondary response. And to recap, what the secondary response is the encounter with the same pathogen, the encounter with the same pathogen again. Okay, so when you encounter the same pathogen again, there'll be a faster increase in your antibody concentration. And this part you can write down in your notes, right? In your notes, you realize that they don't use the word absence of lag period because there's still an absence of lag, there is still a lag period, right? When you see the graph later on, there is still a lag period. So you don't use the term absence of lag period, you use the term faster increase in antibody concentration. Okay, because absence of lag period is theoretically wrong. So why is it a faster increase? Because you no longer have to activate your naive T, your naive B cells, because your memory cells are reactivated easily and they will differentiate into your effector T effector B cells. So then you look at the next one is a higher peak of antibody. So you look at the first two, we use ER terms, we use comparative terms because we want to differentiate between before where there was a lag period and where there was a gradual increase, and we contrast it here with a faster increase. So when you answer questions regarding your primary and secondary responses, and when you're asked to compare them, contrast them, you have to use comparative words, your ERs and your ERs. If you don't use your comparative words, you're not going to get the mark because you don't show understanding that there's a difference. If you just say gradual increase and fast increase, there's no differentiation. You have to use faster, higher peak of concentration. So then the third characteristic of your secondary response is is that your high antibody concentration persists for a longer time. Okay, so if you look at this graph here, okay, if you look at this graph here, we have all the characteristics we just talked about. Okay, and this will be a paper that you all will do later. Okay, so yeah. Okay, so go back. Okay, so next we're going to talk about antibodies. So everything you need to know about antibodies. Okay, so we first talk about its structure. So its structure, we have 4 polypeptide chains. So 2 identical light, 2 identical heavy chains. Okay, and then this is the part where we link it with proteins. Okay, so we're going to talk about 4 polypeptide chains. Immediately, what should come in your head is, what structure does your antibody have? Okay, is it a primary structure, secondary, tertiary, or quaternary structure? Okay, and then we recall in the topic of proteins, when we talk about quaternary, we talk about more than 1 polypeptide chain. So because there are 4 here, so immediately in your mind you should know, okay, it's a quaternary structure. Because if they want to, especially in your paper 3s, in your A levels, that's when they link a lot of topics together, and you have viruses, you have proteins, you have infectious diseases, bacteria, all in one question. So when you all look at your TYS, if you all have started, you all can see that the questions are not just focused on one topic in paper 3. Sometimes even in paper 2, it is a bunch of topics all in one question. Okay, so that's where you have to make those links. So each light chain is paired with 1 heavy chain, and it's held by all 4 bonds. Again, the 4 bonds here, we go back to proteins, and they are your disulfide bridges, your hydrogen bonding, ionic bonding, and your hydrophobic interactions, those 4 bonds. Okay, and within your antibodies, you have variable and constant regions. So if you look at this picture here, you can see all those that are V are your variable regions, and all those that are C are your constant regions. So a common misconception that people have is that this entire portion is variable, but it's not. It's only the upper portion that is variable, and the lower portion, everything below this, everything here is your constant regions. Okay, so that's just a misconception that a lot of people have, and you guys should not have. So next, we have your FAB, your fragment of antigen binding. And then your fragment of antigen binding is highlighted here, where you have FAB. So this entire portion here is your FAB portion. Okay, your fragment of antigen binding. Then next, we have the fragment crystallizable. Fragment crystallizable here is the bottom portion of your antibody. That's your fragment crystallizable. And the last part we have is your hinge region. So the hinge region is Cambridge's favorite region of the antibody, because they always like to test the function of your hinge region, and sometimes they ask you to identify your hinge regions. So you talk about the functions. So the function of your hinge region is to allow your antibody to be flexible. It confers flexibility. So it allows your FAB portion, your fragment of antigen binding, to adopt a wide range of angles, allowing it to bind to antigens that are variable distances apart. Okay, so this may seem very foreign to you, and it's just a bunch of words that don't mean anything. So let me break it down, okay? So we have adopt wide range of angles. This is the first mark. If this is a 2-mark question, this is the first mark. And the second mark comes at the variable distances apart. Okay, you have to state that, because if you don't talk about variable distances apart, there's no reason for it to be able to adopt a wide range of angles. So you talk about the antibody, okay, being able to adopt a wide range of angles. Or if you want to be more specific, the fragment of antigen binding is able, portion of the antibody is able to adopt a wide range of angles. Therefore, it can bind to antigens that are variable distances apart. Okay, so it's really, really important, the function of hinge region, because they love to test it, whether it's MCQ or paper 2, even in paper 3. Okay, so next, a common misconception again that people have is that your fragment of antigen binding is not your antigen binding site. Okay, your fragment of antigen binding refers to the upper portion, the portion that's above the hinge region, if you may, above the hinge region, that is your fragment of antigen binding. But below your hinge region, you have your fragment crystallizable. Okay, and so for one antibody, in this case, your IgG, your immunoglobulin G, okay, it has two antigen binding sites. Okay, I show you in green. So this is the first antigen binding site. This is the second antigen binding site. So the portions in green are where your antigen binding sites, but the portions in purple is your fragment of antigen binding. So you have to differentiate, okay, that your antigen binding site is not your fragment of antigen binding. Okay, next we want to talk about the different classes of your immunoglobulins. Okay, so it's not a must to know what these are, but it's good to know. Okay, and it will help you in your understanding a lot of this topic. So we're just going to focus on your IgG. This is the focus of your syllabus, and we're going to focus on IgD and IgM. Okay, so you have your 5 different classes, okay, of your immunoglobulins. So IgM. So IgM is a pentamer. Remember your monomers? Yeah, but this is 5. Okay, so it has 10 antigen binding sites compared to your IgG that only has 2. Okay, and the difference between your different classes of immunoglobulins is just its functions and its heavy chains. So its heavy chains for IgM, this is the mu heavy chain, gamma, alpha, epsilon, and delta heavy chain. Okay, so um, the functions of IgM you don't really have to know, but it is good to know because if they talk about a pentamer during your exams, you know, okay, it's IgM, and then IgD. So this will help you in your understanding a lot because this was a part where a lot of people got confused when we were studying for infectious diseases. So IgD is an immunoglobulin, but it functions as your B cell receptor. Okay, this will be important later on when we talk about your generation of diversity of antibodies. Okay, this will come in handy later. And just note that, just look at how it is, IgG, how it looks like, and look at how IgD looks like. Okay, we can see that it's very, very similar. But of course, the difference is in its heavy chain. One has the gamma chain and one has the delta chain. Okay, and it serves as the B cell receptor. Okay, your IgD. Okay, then let's talk about active versus passive and natural versus artificial. Okay, so the whole idea of active versus passive is that your active, okay, is that your sorry, is that your active refers to your antibodies being produced by your own immune system. Okay, that's the difference between active and passive. Whereas passive, your antibodies are transferred to you, but your immune system does not participate. Okay, so another difference is that your active confers long-lasting immunity, while your passive confers only short-lasting immunity. The reason behind that is because your no memory cells are being formed when you have your passive um sorry, give me a moment. When you have passive immunity, okay? So active immunity versus passive immunity. One has memory cells formed, one doesn't have memory cells formed. Okay, and it all goes back to the individual's own immune system, if it participates or if it doesn't participate. Okay. Okay, next, natural versus artificial. How to differentiate these two ideas? You can think of natural as something that can happen 300 years ago, and artificial can only happen in 2021. Okay, how I will explain this later. Okay, so we talk about natural active versus natural passive. natural active versus artificial active. Okay, so natural active is just any infection you get. If you get the cold, your body goes through a natural active immunity. And then for artificial is where you get your vaccines. Okay, so you think about it, 300 years ago, they wouldn't have had vaccines. Okay, yeah, they wouldn't have had vaccines 300 years ago. Okay, so then, and why it's active, right, is because both in your infection and in your vaccines, your immune system participates in it. And then that's how your memory cells are formed. That's how you have long lasting immunity. Okay, then we talk about natural passive versus natural active. Natural active, again, is your infection. You get a common cold, you go through your natural active immunity. Natural passive immunity is where your fetus receives your antibodies from your mother via the placenta. Okay, so why it is passive? Because it receives the antibodies, but its immune system didn't go through the innate immune system, the adaptive immune system. It only has the antibodies via the placenta. Okay, it didn't participate in the immune system didn't participate, therefore no memory cells are formed. Therefore, it is naturally passive. Because 300 years ago, fetuses can still receive antibodies via the placenta. I know it sounds weird, but when you go into your exam, you remember 300 years and then it will help. Okay, so artificial passive versus artificial active. Artificial active, your immune system participates, therefore memory cells are formed. But artificial passive, your immune system doesn't participate, and uh, it didn't exist 300 years ago. Okay, so artificial passive, an example of it would be anti-serum. So this anti-serum you receive from another person that has antibodies. Okay, so you think about it again, okay? So 300 years ago, you didn't have it and your body didn't participate in the immunity, okay? So that's how you differentiate, because some people um, have a difficult time differentiating between these four concepts, okay? So I hope it's helpful, the 300 years thing, okay? Okay, here we will have a quick 5 minute break. You can go and drink water. Okay, 3 minute break. You go and drink water, and then you'll come back. We'll start again at 2041. Okay, because this is gonna be a lot. So you'll take a break first. Okay, there are questions in the chat. Huh. Okay. What is the significance of FC region? B cells. Okay. Oh, most common topics that. Okay, wait. Let's start from the top. Okay. Okay, so the most common topics that this can be linked to would be bacteria, viruses, because it's the most linked, no? It can also go to proteins and yeah, they could literally link it with anything they want to. They could even link it with cell signaling, right? And then they give you a cell signaling pathway. They can honestly link it to anything. But cell signaling is a bit far fetched. But that would be the worst combination, no? Cell signaling plus infectious diseases at LJ. What does a T cell and B cell do? I hope I went through that, right? What does a T cell and B cell do? Okay, and
No,
are B cells?
I just sent a new message, so it's all the questions
Okay.
there already. Yeah.
Okay. Okay, thank you. Okay. What does a T cell and what are B cells APCs? B cells are not APCs. APCs are only your dendritic cells and your macrophages. Okay. Significance of FC region. Okay. If you this part I'm not gonna talk about in the lecture in the lesson today. Um, okay. So it is used in opsonization. Opsonization is how your antibodies work. Okay, I can talk about that. Should I talk about it now? Okay. Okay. Yeah, we can talk about it now. Okay. Opsonization. Let's see if it's here. Opsonization. Okay. Okay, so it's not here. So okay, someone asked about the significance of your FC region. So I'll just talk about it really quickly. Okay. So there are three ways your antibodies work. Okay. Not all three will be taught in every school, but okay. So the first one we have is opsonization. This is still relevant to the topic, so I'm going to now and not after the lesson. Okay. So opsonization refers to your binding of antibodies AB to your pathogen. Okay, and then it tags the pathogen for phagocytosis. Okay, so how it tags it, right, is so the FAB portion binds to your antigen, while your FC region is recognized by phagocyte. Okay, you don't have to know how it's recognized by the phagocyte, but you just know that the FC region of an antibody, a function for it is that it can be recognized by your phagocyte to promote phagocytosis so that pathogens can be removed via opsonization. Okay, I'm not really going to talk about how these, I'm not going to talk about these three things much because it's more of how to understand the topic better. Okay, but it's not really the core syllabus. So second one, we have agglutination. And the last one we have is your neutralization. Okay, so you just need to know the last two, but opsonization just refers to your FC region of your antibody being recognized by your macrophage. Okay. Yeah. Okay. So we go back to the lesson. I'm sorry that took a little while. Okay. Okay. So okay, let's come back to the lesson. Okay. So this is gonna be a lot. Okay. So you'll just bear with me. Okay. Okay. So now we're talking about the mechanisms to generate antibody diversity. So there are three mechanisms, and you have to know all three by heart. And it seems really daunting that there are three, and it seems really daunting the idea of antibody diversity, but it's really, really, really simple if you properly understand it. Okay. So before we dive into the first one, we talk about what our uh, heavy chain and light chain genes contain. Okay, so we'll talk about the heavy chain first. Your heavy chain gene consists of 4 gene segments. You have your V, D, J, and C gene segments. Okay, 4 gene segments on your heavy chain. You can think of it and you can understand it as because there are 4 gene segments, therefore there's a lot, and there's a lot, therefore it is heavy. Okay, light chain, because it's lighter, it has one less, it only has VJC. And people from VJC should remember it very easily because, VJC, right? Okay, so your multiple gene segments, right? So your VDJC for your heavy chain gene segment and your VJC gene segments for your light chain gene, the multiple gene segments all contribute to a vast number of heavy and light chain diversities. Okay, so this is the basis of your somatic recombination, which is your first mechanism to generate antibody diversity. So somatic recombination, another name for it is your VDJ recombination or your VJ recombination. Okay, so the function of somatic recombination is so that your multiple VD and J gene segments can allow for the generation of many antibodies, and each of those many antibodies have a unique antigen binding site. Okay, so the whole function, simplified function of somatic recombination is just to generate a lot of antibodies, and each of these unique antibodies have a unique antigen binding site. That's the whole function of somatic recombination. Okay, and this occurs in your developing B cells. Your developing B cells are have not been okay. So is before your naive B cell even comes about. Okay, is while it is developing. Okay, so we're talking at the gene level here, in case you need clarification. Gene level. So we talk about the DNA strand. Okay, so on your heavy chain, for somatic recombination, your heavy chain, you're talking about two steps and only two steps. Okay, first step, one D gene segment rearranges to one J gene segment. Okay, so the keyword here is rearrange because you talk about recombination. So one D gene segment rearranges to one J gene segment, forming a DJ rearrangement, which is here. And then your second step is where your one V gene segment rearranges to one DJ rearrangement. Okay, so it seems really confusing now. So I give you a picture to better visualize it, okay? So, okay, this is your somatic recombination for your heavy chain. So on your germline DNA, or just basically DNA, okay, you have multiple gene segments, like I said just now, your V, D, J, and C gene segments. So your first step is where your D and J join together, or where they rearrange together. So one D, one J, and this one here, and this one here, they rearrange to form your DJ recombination. Okay, and then the second step is where your V rearranges to a DJ rearrangement. So there is this step where this V gene segment rearranges with your DJ rearrangement to form your VDJ rearrangement. Okay, so let's link this back to our topic of DNA and genomics, or whatever your schools call it. Okay, so the topic on transcription and translation and replication. Okay, so your DNA is being rearranged first. So after your DNA is rearranged, it has to go through transcription so that your mRNA can be produced, so that your mRNA can be translated, so that you can have your B cell receptor. Okay, so after your VDJ rearrangement has occurred, you have to go through transcription. So after transcription, we have our mRNA. So this is your RNA level. Okay, and then after it transcribes, you form this whole thing, which is your VDJ plus a constant gene segment. Okay, but now they're called exons because we're talking about it on the RNA level. So your VDJ exon and your C exon are now translated to form your protein. And this protein is your BCR, your B cell receptor. Okay, it seems like a lot. I will repeat. Okay, so we talk about heavy chain gene segment. Heavy chain gene segment made out of B, D, J, and C gene segments, 4 gene segments. So your first step of VDJ recombination, so your somatic recombination for your heavy chain, the first step is to form a DJ rearrangement. So 1D to 1J give you 1DJ. Okay? Then 1V to 1DJ gives you 1VDJ. That's why it's called VDJ rearrangement or VDJ recombination. Okay, so after your VDJ recombines, okay, transcription occurs, your pre-mRNA, okay, this is the keyword, right? Pre-mRNA. Why it's called pre-mRNA is because it contains your other portions as well. If you look at this picture here, okay, you look at right after transcription, so this point here, right, the primary transcript. So this is another word for pre-mRNA. Your primary transcript, you can see that it still has other V gene segments. It still has other J gene segments. Okay, you have to get rid of all those. You have to get rid of all those. So you undergo RNA splicing. RNA splicing brings your VDJ recombination. So this portion here, it brings it beside your C gene segment. Okay, and why does it bring beside? You don't use that in your exam. It's just for you to understand. Okay, so you bring that beside your C gene segment, so that you now have your all 4 gene segments of your heavy chain to be ready to be translated to give you your BCR, your B cell receptor. Okay, some people might ask, why is it the B cell receptor and why are we not talking about the heavy chain? Okay, so remember just now when we were talking about the different classes of antibodies. Okay, the different classes of antibodies. So let's look at it, okay? The final product we want to get, the final product is this IgG. Okay, this is the aim of the game, okay? But now what you have is your IgD. This is what you have at your cell surface membrane of your B cells. So we're talking about the heavy chain. We're talking about specifically this one portion here, the purple portion that I circled. This one created after 1 times VDJ recombination. Okay, I repeat. Okay, so you have 2 heavy chains on your B cell receptor. One of it is made after 1 round of VDJ recombination. Okay, so after that, you do 2 rounds, you get 2 heavy chains. So 2 heavy chains, and then you do 2 rounds of your VJ recombination, then you get your 2 light chains, after which you get your whole B cell receptor. After everything, after the activation of your B cells and differentiation of your B cells, your B cell receptors will undergo class switching to give you your antibodies. You don't have to understand the last part. We'll go back to the last part again later. So just understand VDJ recombination. If you go through 1 round of that, you get 1 round of your heavy chain. Okay, because just now we're talking about 1 chain being 1 polypeptide chain. 1 heavy chain is 1 polypeptide chain, but you have 4 chains here. 4 polypeptide chains gives you 1 immunoglobulin G, immunoglobulin D monomer. Okay, if you have any questions, just drop it in the chat again. We'll answer that later. Okay, so that's for your heavy chain. Now we go to the light chain. The light chain is the exact same thing, just that you have 1 less step. So it is basically 1 step, okay? Your VJ recombination is 1 step. So you have your DNA rearrangement of just 1 J gene segment. Let me use light blue. 1 J gene segment. 1 J gene segment here to 1 V gene segment here. So both of them come together to give you this VJ recombination here. Okay? So after you get the VJ recombination, the exact same thing happens. You go through transcription. Then you get, now we're talking about the RNA level. Okay, you go through transcription, you have your pre-mRNA, your primary transcript. Your primary transcript has to go through RNA processing. The specific term is RNA splicing. So it goes through RNA splicing. So now your VJ exon and your C exons are joined in your VJC. Okay, here. And then after this, you get your mature mRNA, and your mature mRNA undergoes your translation to give you your protein. Okay, you can ignore the last part. Okay, it gives you your protein, and the protein here that we're talking about is your 1 light chain, 1 light polypeptide chain. Okay, so you have to go through 2 rounds of your light chain recombination, 2 rounds of your heavy chain two rounds of your heavy chain recombination to get one IgG or one IgD monomer. Okay, so we recap again. Your light chain recombination occurs in one step. One V gene segment, okay, rearranges to one J gene segment. That's it. Okay, after your VJ recombination, same thing as your VDJ. You get transcription. Transcription, you get pre-mRNA. Pre-mRNA undergoes RNA splicing. RNA splicing, your VJ and your C exons are joined. Then you go to translation. Translation gives you the polypeptide. But if they're talking about somatic recombination, they are stopping at here. Okay, they're just talking about the first two steps for your heavy chain, and they're only talking about one step in your light chain. That's all they're talking about for somatic recombination. But to understand how this generates diversity in your antibodies, you have to understand the transcription, the translation, and between the two, you have your pre-mRNA undergoing RNA splicing. Okay, so yeah, that's how you think about infectious diseases with your transcription, your translation, and you can recap your post-transcriptional modifications. And you study. So when you study bio, you don't study one subject by itself, one chapter by itself, but you study it together. Okay, so an example would be like this, or if you want to, you study your transcription, translation, replication together with your modification. So for RI students, you will have DNA genomics 1, DNA genomics 2, and pro and eukaryotic 2. So you should study all 3 chapters together if you are from RI, because that's the syllabus. I don't know about the other schools. Okay, I'm sorry. Okay, so next we have the second mechanism. Second mechanism is a little tougher, okay, but it will make a lot of sense. Okay, so the second mechanism, somatic hypermutation. So hypermutation, its name suggests that mutation is occurring at a very fast pace, at a very fast speed, okay? And then the function of somatic hypermutation is to further diversify, okay, not to diversify, but to further diversify your variable regions for antigen binding. Okay, what does this mean? Okay, after your VDJ recombination, you have, after your somatic recombination, you have your many antibodies that are generated already. Each has a unique antigen binding site. Okay, but then after you've generated all those antibodies, now you have to make sure that they are specific to and they are very complementary to. Again, don't use this word in your exams, just for you to understand. So that they are very complementary to, so that their shape, right, is more complementary to the shape of the epitope. The conformation of the antigen binding site of your antibody is more complementary to the antigen conformation. Okay? So how this happens is, it's a bunch of point mutations. So your point mutations occur during your clonal expansion, or in other schools, during your mitosis, right? At your rearranged VDJ or your rearranged VJ regions. Okay, so these point mutations, because we learn in DNA genomics, your point mutations in your DNA can code for, not will code for, but can code for different amino acids. So if you want to link it again, I'm sorry I keep talking about linking to other topics, but if you link it back, you can talk about your missense mutation, your nonsense mutation, and all those different things. Because your missense mutations, your nonsense mutations, silent mutations, all of these are all going to contribute to whether or not your amino acid changes, or whether or not a functional protein is even produced. Okay, so your slight amino acid differences in the variable regions of your Ig chains, on your immunoglobulin chains, okay, they will change the entire conformation of your antigen binding site of your antibodies. Why? Because after your amino acid changes, they have different characteristics. Each amino acid has a different characteristic. So the bonds formed and the conformation after the different bonds are formed changes. So link back to proteins, your 4 interactions. Now, the conformation of your antigen binding site has changed. Therefore, some mutations result in your antibodies having a higher affinity. Some of it will result in it having lower affinity to your antigens. So I recap from the start. Somatic hypermutation just means a lot of point mutations happening very quickly. These mutations will lead to amino acid differences. Amino acid differences will lead to whether or not the antibody has a higher affinity for or a lower affinity for your antigens. That is the whole idea of somatic hypermutation. Okay, so after this, your B cells that express higher affinity. Okay, so now we use B cells. B cells because we're talking about the B cell receptors on your B cells. So your B cells that express higher affinity for your antigen are selected for clonal expansion and differentiation. You can think of it like your evolution. When we talk about um, favorable characteristics, they are selected for, and then they are allowed to breed. So this is a similar concept. It's not the exact same concept. It's not natural selection working out here, but it's similar to. That's how you can understand it better. Okay, B cells that have higher affinity. Okay, they are selected for clonal expansion and differentiation. This term, okay, the term for this whole process in this line, okay, is called affinity maturation. Again, your school may or may not use this term, but this is what this term means. Your B cells that have high affinity for the antigens that it is specific for are now selected for clonal expansion and differentiation. Think of it logically. If I have a B cell that has a B cell receptor that is not so complementary to my antigens, why would I want so many of it? Should I not wait for a B cell that has a B cell receptor that is almost 99% complementary to my antigen, so that if I produce so many of this, it is more effective in getting rid of the pathogen inside my body that I do not want. Right? Of course, you want an antibody that has higher affinity for your um, antigen that you don't want in your body. Okay. So now you might get confused because I talk about B cells, I talk about B cell receptors, and then I talk about antibodies, and then I just use these terms all interchangeably while explaining. So what do I mean? Okay, so I have a question for everyone here. So just now we were talking about B cells, right? Here we're talking about B cells here, right? Specifically. So why are we talking about B cells here, and why are we not talking about antibodies? Okay, you all have what, 2 minutes? Okay, you have 2 minutes to go and think why I'm talking about B cells here and why I'm not talking about antibodies here. Okay. Yeah, you can just write it down if you want to. Okay. So okay, so Okay. So the answer to this is because just now we were talking about our IgD and IgG. So your IgD, we refer to them as B cell receptors, right? Because they serve as your B cell receptors. That's its function. Okay. So your IgDs are on your B cells, and your IgGs are what we want to produce. So when we talk about B cells here, we're talking about your IgDs, and we're talking about your IgGs. Okay. So the antigen binding site of your B cell receptor or your IgG, your IgD, okay, I should write it down here. Your IgD is equal to the antigen binding site of your IgG. Okay, they are just different classes of antibodies, and the difference between your different classes of antibodies, like we talked about just now, is just your constant region. Okay, is your constant region. And because of the difference in constant region, they have different functions. So your IgG, its function is an antibody. Is to agglutinate, is to opsonize, is to neutralize your pathogens. But the functions of your IgD is to serve as a B cell receptor. Okay, so class switching is the easiest of the three mechanisms to understand. Class switching just means one thing. Your variable region of your heavy chain, okay, the variable region that we generated just now during our VDJ and our VDJ recombination is now rearranged to a different constant gene segment. That's all class switching is. One thing, variable region now is connected and now is rearranged to a different constant gene segment. That is it. Okay, so this occurs at your heavy chain gene locus. Okay, so a question, just to make you think. After VJ recombination, will there be will it undergo class switching? Okay, the answer is no, because the I don't want to say caveat, but the thing to pay attention or the thing to realize about class switching is it only occurs in your heavy chain. Only in your heavy chain. That's why I wrote heavy chain here. It does not occur in your light chain. Only in your heavy chain. Okay? So so we recap the three mechanisms because it's been a lot. First mechanism, VDJ recombination, somatic recombination. Okay, this just its function is just to generate as many antibodies as possible because you have multiple V, multiple D, multiple J, multiple C gene segments. You want to generate as many antibodies as possible. Why? Because you want as many unique antigen binding sites as possible so that any pathogen that comes into your body, you can get rid of it. You know, like COVID, we have never had SARS-CoV-2 in our body before, but we don't want to get caught off guard. You know, so we want to generate as many antigen binding sites as possible so that by chance we can get one that is complementary to your SARS-CoV-2 so that you can get rid of it fast. Okay, so your that's your somatic recombination. Something to take note of is that it occurs during the development of your B cells. Okay, before your B cells have even been properly formed, before your naive B cells have even been formed. Okay, for somatic hypermutation, we're talking about activated B cells. So after they have been activated already. Okay, so after your naive B cell has encountered the antigen, so it already has the peptide MHC complex on its cell surface membrane. And after the helper T cell has recognized that peptide MHC complex on the naive B cell's cell membrane, and it gets activated. After it gets activated, it already has the B cell receptor on its cell surface membrane. Now, then it undergoes somatic hypermutation. So it undergoes somatic hypermutation. You get is basically getting a selecting for B cells that have higher affinity for your antigens. That is the whole idea of somatic hypermutation. So now, B cells that have high affinity undergo mitosis, undergo differentiation, so that you can produce as many as you want of the plasma cells, so that the antibodies that they secrete have high affinity for your antigen. And how come the B cell receptor and the antibody have the same antigen binding site? It's because the only difference between these two is a constant region. Okay, one has a gamma chain and the heavy chain. One has a delta chain and its heavy chain. If you don't understand it, I'm referring to the picture of the different classes just now. Okay, so that is somatic hypermutation. Now, class switching. Class switching is the easiest of the three. We're just talking about heavy chain. What happens at the heavy chain? The variable region that was already generated just now during your somatic hypermutation is now connected to a different gene, a different constant gene segment, so that you get a different class of antibody, so that it can serve a different purpose. So your IgD will now be your IgG. It could now be your IgM. It could be other classes of immunoglobulins, so that you can continue with the whole immune system. The next process can begin. Okay, so is a lot. Just remember that class switching is the easiest, and somatic hypermutation is just a little complicated. It's not too complicated. It's a little complicated, and you're done. Okay, that is diversity of antibodies down. That's the hardest part of the entire subject chapter. Okay. Next, we're going to talk about antibiotics. So there are two types of antibiotics. We have bacteriostatic, bactericidal. Okay, so bactericidal, you can think of it as killing, because homicide, killing, and static just means they're not moving. So bacteriostatic antibiotics just keep the bacteria at its state so that it cannot reproduce. Okay, so that is antibiotics. So for antibiotics, we're going to focus on okay, so antibiotics, we're going to focus on penicillin. Penicillin is the focus of the syllabus, and it is very, very simple. Okay, so in order to understand how penicillin works, you have to understand how a bacteria undergoes binary fission. Okay, binary fission, we recap, is just cell division, but for bacteria, so that they can reproduce. Reproduce not the correct word, but so that they can produce two bacterial cells from one. Okay, so how it undergoes binary fission is its cell wall has to be broken down and reformed. Okay, it seems a little foreign, but okay, the bacterial cell wall is made out of peptidoglycan. Peptidoglycan has to be remodeled during bacterial growth, which is the first step. Okay, it has to be remodeled. And how it is remodeled is the cross links, okay, that are between your peptidoglycan chains, they have to be broken first. After they're broken, they're reformed. Okay, and when they are reformed, is when you have a longer chain or a extension of your bacteria cell wall. So the theory behind it is it has to break, then it has to reform. Okay, so transpeptidase, this is an enzyme because of the ASE at the back of the word. Transpeptidase is the one that reforms these crosslinks. Okay, so if we recap, all enzymes have an active site. So transpeptidase has an active site. It is complementary to your peptidoglycan chains, or portions of your peptidoglycan chains, so that they can reform the cross links. But it is also complementary to penicillin. Okay, so penicillin is a competitive inhibitor. And if competitive inhibitor sounds foreign to you, recap your enzymes topic. It is a competitive inhibitor. It binds to your active site. Okay, so if it binds to your active site, now your transpeptidase is no longer able to reform your cross links. So if it is no longer able to reform your cross links, right, the net effect on the bacterial cell. It's just that the peptidoglycan chains are just gonna be broken. Okay, it's just gonna be broken. That's the net effect, because usually it's broken and it's remodeled, broken, remodeled. Net effect, nothing, right? Net effect is you get more. But now you're just breaking it and you're not remodeling it. So the net effect is that it's gonna create, or it's not gonna create, but it's gonna lead to a weaker cell wall. Okay, so now the cell wall is weakened, and after it is weakened, when the bacterial cell is taking in water by osmosis, the turgor pressure within your bacterial cell increases, and because now it's a weakened cell wall, your bacterial cell will swell and eventually it will lyse. Okay, so penicillin mode of action, what you talk about is you talk about how your cell wall is broken and remodeled. Then you talk about what enzyme causes this. Transpeptidase and its active site. Okay, because that is the whole basis of how penicillin can work. Is that its active site is complementary to penicillin. Penicillin is a competitive inhibitor, binds to the active site. Therefore, peptidoglycan cross-links cannot be reformed. And because it cannot be reformed, it is a weaker cell wall. And when you take in water by osmosis, the turgor pressure increases against your weakened cell wall, causes it to swell, and eventually, because it's weak, it will lyse, and when it lyses, it dies. Okay? So that is penicillin. And this is the last thing for today. Okay, tuberculosis. Tuberculosis is it's a little difficult. Okay, so the mode of transmission, okay, is very similar to COVID. If that will interest you to study the mode of action of mode of transmission of TB. Okay, so it's transmitted via aerosol drops. Is translated via fine aerosol drops like COVID when an infected person sneezes or coughs. Okay, and therefore it is an airborne disease like COVID. And okay, so how it is, how does it infect your body? Okay, so once it's inside your lungs, the bacteria, okay, Mycobacterium tuberculosis is a bacteria. So when this bacteria is inside your lungs, okay, your alveolar macrophages. Alveolar macrophages just mean your macrophages in your alveoli. They phagocytose the bacteria and form phagosomes. So if you are unsure about the process of phagocytosis, go back to the top and familiarize yourself with it. So it goes in the phagosome. So the next step of phagocytosis is it has to fuse with a lysosome, okay, to form a phagolysosome. So let's assume that happens. So your phagosome fuses with your lysosome, now forms a phagolysosome. But the natural next step in phagocytosis is it now has to digest the bacteria. But the special thing about Mycobacterium tuberculosis is that it has a thick mycolic acid cell wall. This thick mycolic acid cell wall protects the Mycobacterium tuberculosis, protects the bacteria from the reactive oxygen species and your acids inside your lysosome. Okay? Or sometimes we refer to them collectively as your hydrolytic enzymes. Okay, so that is scenario one, where the phagosome fuses with your lysosome. Scenario two, your Mycobacterium tuberculosis inhibits diffusion. Okay, so the natural step is for phagosome, lysosome to fuse to form your phagolysosome. But now it can no longer form because your bacteria inhibits it. You don't have to know how it inhibits it, just know that it can inhibit it. So after it goes, okay, so we recap. Okay, the bacteria goes into your lungs. Your macrophages phagocytose your bacteria, forms your phagosomes. Two scenarios can happen. First scenario, phagosome fuses with your lysosome, but your bacterial cell wall, because it has a thick mycolic acid cell wall, it protects the bacteria from the reactive oxygen species and acids. It protects the bacteria from hydrolytic enzymes within your lysosome. Second scenario, your bacterium inhibits the fusion of your phagosome and your lysosome. So there's no phagolysosome. So again, your bacteria is not exposed to your hydrolytic enzymes in your lysosome. Okay, so because no phagolysosome is formed, no lysosomal enzymes can kill the bacteria. Next step, the bacteria survives because it is not killed by your hydrolytic enzymes. It survives. It survives. It continues to multiply inside your macrophage. It reproduces within your macrophage. Okay, so now there are many bacteria within one alveolar macrophage. Okay, now other macrophages, other than other macrophages, other dendritic cells, and your lymphocytes. Lymphocytes again, your T cells, your B cells. They form a tubercle. What is a tubercle? Tubercle, it functions to wall off. Okay, to wall off your bacteria so that it cannot spread outside of this wall. Let me show you in the picture. Okay, the big mass here is a tubercle. Okay, inside your tubercle, before it became a tubercle, you had your bacteria here. You can't really see that. You have your bacteria here. Okay, but and your bacteria is inside. Your bacteria is inside the macrophage reproducing. Problem is you don't want the bacteria to reproduce so much, and you don't want it to be able to go to other parts of your lung because you don't want it to infect other parts of your lung. So what does the body do? It forms a tubercle. Tubercle is made out of your lymphocytes, your dendritic cells, and your macrophages. It just forms this wall-like structure here to wall off the bacteria. Okay, so next step. At the center of your tubercle, in the center of the mass, your cell, which is your macrophage, or other cells in your alveolus, will start dying because they've been infected by your bacteria. They start dying, but this time it doesn't die, or the cell death is not by apoptosis. Cell death is now by necrosis. If your school doesn't make a distinction, you can just use cell death generally. Okay, and after your cell dies, okay, the cell membranes will rupture, and after they rupture, the cell contents, including your bacteria, is now spread to your surrounding. So you can look at it here again. So what happens is, at the center of your tubercle, also known as the tubercle cavity, your macrophages die by necrosis. So when they die by necrosis, your cell membrane is ruptured. And when it's ruptured, the Mycobacterium tuberculosis, the bacteria that's inside the macrophage, that was inside the macrophage, is now released to the surroundings. And it can now infect more and more and more and more. Okay? So it can either continue infecting more or it can stay latent. Okay? So let's assume latency. So latency, nothing really happens. And then afterwards, suddenly, chance event, right? Your tubercle cavity enlarges. So now that your cavity enlarges, you have more bacteria inside, more bacteria, more infection, and then more cells die by necrosis, more bacteria form. It's just a snowball effect, okay? So now you have an air-filled cavity at the center of your tubercle so that your bacteria can multiply outside of your macrophage. It no longer has to infect a macrophage, no longer has to infect a cell to be able to multiply. Cancel last. It can no longer infect a macrophage so that okay. It can no longer infect your macro. It no longer has to infect your macrophage to be able to multiply. Okay. Then your tubercle eventually ruptures, and when it ruptures, then your bacteria is just going to go everywhere. So this is the rupturing of a tubercle here. The rupturing of a tubercle here, and then you can see that the air cavity has enlarged from before, and now so much bacteria is now allowed to go into your bronchioles. okay, into the rest of your lungs to infect even more parts of your lungs. Okay, then a cough will eventually develop, and after it develops, uh, after the person coughs, it facilitates the aerosol spread of the bacteria, and then your lungs progressively get destroyed. If you want to scar yourselves, you can go and Google tubercles in lungs, and then you can see the holes in the lungs, and then the holes in the lungs are your airfield cavities. So um, that is it for TB, and that is it for the entire, not entire lah. Okay, we didn't talk about vaccines, but the majority of infectious diseases. Okay, now I will give you all a short paper. Okay, it's very, 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 very simple. Okay, okay, it's just a recap for today. Y'all can just write down on a piece of paper beside you. Okay, I will send these to the chat. I'll send these to the chat. Okay, then you can refer to them. Or y'all could go for a break. You can choose to do either. Okay, um 15 minutes. Okay, that's not 15. You have 14 minutes, okay, until 21:40 to do this paper, and 14 minutes is more than enough. Yeah. Okay, it's in the chat, so you can look at that and do them if you want to. Okay.
Is it like they recruit while they have new people? What number is that? What are you?
Okay. Hello. Okay, so someone pointed out a mistake. So in just now when I went through, I said the correct thing, but I wrote the wrong thing. So you can hear me, right? Um, yeah, okay. Um, yeah, I said the correct thing that the for class switching, the constant region is changed. The variable region is the part that is unchanged. But just now I wrote something. I wrote the wrong thing. Okay, so constant region is changed, variable region remains unchanged. So thank you, Jay. Okay, so the paper. Okay, I will send the answers to the chat first. Okay, so the paper is okay. Okay, we'll answer KP's questions later. Okay. So the paper is a typical infectious disease paper. The only thing that is that may not be very similar to A levels is that it is not cross topic, or it's not. Yeah, it's not cross topic at all. Yeah, this is a purely infectious disease question. So again, in the A levels, it'll be very, very cross topic. Okay, so we go through the first page. You should get all correct now. Huh? Right, it's quite easy. It is okay, of course, if you leave out the cells here, because they wrote cell, and it's of course okay to leave out response because they talk about response. Another thing is there are two answers for response 2. There is the adaptive immune response and there is the cell-mediated response. Both are accepted. Okay, it's just that the cell-mediated response would be the more appropriate answer, would be the more correct answer. Okay, say the structural difference between IgM and IgG. This is very, very simple. So another answer, right, that is accepted for structural difference between IgM and IgG is IgM has 10 antigen binding sites, IgG has only 2, or IgM exists as a pentamer, IgG exists as a monomer. Those are also accepted answers. And then, of course, remember that you have to specify the word heavy because because class switching only refers to the changing of the constant gene segment at your heavy chain gene locus, only at your heavy chain. Okay. Next we have this one. Okay, so this is a very, 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 very popular kind of question. Not this specific question, but this kind of question. So, firstly, when you talk about the describing of differences, you have to state what the differences are, and you have to quote your values to substantiate your point on the difference. Okay, so and quoting values is very, very important. You don't quote your values, you lose 1 mark, you lose 2 marks sometimes even, or you don't get marks at all, depending on your teacher. But in the A levels, of course, you only minus 1 mark. Okay, and so the difference between your concentrations of IgM and IgG from day 10 to 30 is that your IgM peaks earlier. Okay, so it peaks earlier at day 13, 14, while IgG's concentration peaks at day 21, 22. Okay, is you don't write 21/22 in your answer sheet, but that I'm just giving you the range of values that is accepted for as the answer. Okay, and so what I did here was the opposite of what I wrote. So I stated after I quoted the values. I mean, either way works as long as you have both in your answers, okay? And because you talk about differences, you have to use comparative words. Otherwise, your you have to use comparative words. Otherwise, your answer doesn't make sense. Okay, so you have to use comparative words like higher, er. Okay, when you talk about differences. Next, you have your two differences. Okay, so there are more than two differences. But when you see this question, it should immediately trigger your thoughts about two things. Firstly, higher peak concentration. That should be the first thing that comes to your head. The second thing, your shorter lag time. And then notice that when you think about the differences, because we talk about differences again, you have to use your comparative words ER and ER, because if you don't get the marks. Okay? So your first difference, again, you quote and you use comparative words. You quote the numbers. So the lag period is 11 to 12 days in the primary response, while it's a shorter 4 days in the secondary response. The explanation, of course, is because memory cells were not present during the primary response, but they were present in the secondary response. And then the memory B cells, they reduce the lag period because they take a shorter time to differentiate into plasma cells. Okay, there was a question in the chat just now that asked why memory B cells take a shorter time to be activated compared to your naive B cells or your memory T cells and naive T cells for that matter. So you can think of it this way. You think of the response as a progression. So level 1, level 2, level 3, level 4. So you have your level 2, which is your naive T cells, your naive B cells. Level 3 is your effector T, effector B cells. Level 4 is your memory cells. Okay, so your memory cells do not have to go through level 1, which is your antigen presentation. it doesn't have to go through antigen presentation to your naive B cells, your naive T cells, and they don't have to, and after that they get clonal expansion. So it doesn't have to go through antigen presentation. It can just immediately get reactivated. Don't have to know how it gets reactivated. Just know that it can get reactivated to differentiate into your effector effector cells, which include your plasma cells, your helper T cells, and your cytotoxic T cells. Okay, don't have to know how because that's out of the syllabus. If you want to know, you can go and Google it. But it is out of your syllabus. Just know that it takes a shorter time to differentiate into your plasma cells because 4 to 3 is way faster than 1, 2, then you go to 3. Okay, think of it that way. It will help, I guess. But if you really want to know, you can go and Google the answer, or you can go and ask your teachers, approach your teachers for the correct answer. So the second difference, so the first difference we're talking about the reduction in lag period. Second difference we're talking about is the peak concentration differences. So your peak concentration for your immunoglobulin G reaches a much higher concentration in the secondary response compared to your primary response. So again, we are talking about comparative words. And okay, so some of you might ask, why am I not quoting the numbers? Because if you look at the graph, no numbers were given on the, this is the, I want to say y-axis. Is this the y-axis? I have completely forgotten, but it's the vertical axis, okay? So the vertical axis has no numbers. So then you can't quote, so it's okay. They don't expect you to quote, of course. So you just write the comparative word higher. Okay, so now we have the explanation as to why it reaches a higher concentration. So the pool of memory B cells will differentiate to form more plasma cells, which secrete more antibodies in the secondary response, increasing the concentration of your immunoglobulin G of your antibody. Okay, so this is I didn't cover this in the lesson, and they don't usually talk about this in class, but just understand that during your secondary response, you have more B cells. Again, you don't have to know why. So this is the problem with the syllabus. They teach you that it's more, they don't tell you why. Okay, so you just know that there's more. Okay, so you have more B cells now. You have more plasma cells now because your pool of memory B cells can differentiate to form more plasma cells, which secrete more antibodies. So the comparative word here would be more. And you use that to explain. Okay, if you want to know why, again, you can go and Google, you can go ask your teachers, but it's not part of the syllabus, which is very, very weird. Okay, so I hope you got that. Yeah, so again, just comparative words. These are the exam remarks, by the way, the ones on the right. Yeah, so you talk about the presence and absence of your memory B cells in your secondary and primary responses, respectively. The last, this is the last question, right? The last question is so simple. Describe how your plasma cells can produce different types of antibodies. So this just immediately should just trigger class switching. Then you look at the marks, 2 marks. So class switching, this is 1 mark. Explain that it is due to class switching. Then explain that, and then you explain the process. What's this called now? What is class switching? Okay, so you just say that it is class switching, and you just give a very brief explanation because it's just 2 marks. You know. Okay, so that is it for the paper. So now we will have I'll just finish up the questions that are in the chat, and then after that we will move to Steffi. Okay, I just finish up the questions in the chat. Okay, so I hope I answered the question on why activation of memory T cells is faster. We're talking about the levels 4, 3, 2, 1. Then we're talking about the when are they considered mature, when are they considered specialized is after differentiation. Okay, specialized, of course, after differentiation. Matured is also after differentiation. So I hope that was that. Okay. For class switching, okay, yeah, this was the mistake that I wrote. How does it rearrange to form a D and J rearrangement? It's not thing, it's rearrangement. Okay, so how does it rearrange? Again, not part of the syllabus. Just know that it is rearranged. I'm not trying to Skype, I'm not trying to cheat you of the answer. It is really just not in the syllabus, not in our syllabus. Just know that it is rearranged. Okay. Yes, thank you so much. Yeah. Okay.